Hello everyone, how are you all? I hope you are doing well. This is Dr. Ekta and today we are going to understand what chromosomes are and how important they are for us. And the main thing which I will be discussing in this video is how to recognize chromosomes or karyotyping. Okay, I will be sharing the presentation with you. Okay, let's move forward. Now, what is karyotype? Karyotype is a test to identify the chromosomes and evaluate their structure and number and make a karyogram. Now question comes, what is karyogram? Karyogram is nothing but a graphical representation of chromosomes in a cell wherein chromosomes are organized in pairs and numbered sequence according to their size, that is from larger to small, and the position of centromere. This is a human karyogram. Most of us know humans have 46 chromosomes or 23 pair of chromosomes. Out of them, 22 are autosomes and one pair of sex chromosome, that could be XX or XY. So, standard nomenclature for human karyotype is 46XX for females and 46XY for males. So by karyotyping, we detect if there is any deviation from this standard nomenclature, means if there is any change in structure or number of chromosome. If that is the case, that means there may be an abnormality in that particular person of which we are seeing the karyogram. For example, in Down syndrome people, normally we have 2 copies of chromosomes, but in Down syndrome patients, there are 3 copies of chromosome 21 are found, that is also called trisomy 21. So we understood how important is to identify chromosome. Okay. Okay, that was about the number. Let's move forward with the morphology of chromosome. Chromosomes are thread-like structure which is made up of DNA and histone proteins. These proteins are wrapped around the DNA. As we can see here, this is the DNA and these are the histone proteins which are wrapping this thread-like DNA to form nucleosome. Nucleosomes are nothing but a structural unit of chromosome which is made up of DNA and protein. This structure is important. Why? Due to this unique structure of chromosomes, DNA is able to fit inside the cells. For example, if all the DNA molecule in a cell were unwound from histones and placed end to end, they would extend up to 6 feet. So this packaging is crucial. So number and structure of chromosomes both should be in exact certain way. Okay, this is the chromosome and these are the chromatids. Chromatids are the identical halves of each other which are joined at centromere. These are the 2 arms of chromosomes, P arm that is smaller and Q arm that is the longer arm, and this is the terminal part which is called telomere. Okay, in different organisms, chromosomes vary in number and structure or shape or size, anything. Like most bacteria have 1 or 2 circular chromosomes, while humans along with other animals have linear chromosomes. Like in humans, as we discussed earlier, there are 46 chromosomes in total or 23 pair. Each pair contains 2 chromosomes, one coming from father and another from mother. As we are diploids, we have homologue of each chromosome. Let's say this is chromosome 1 and these are the homologue. So this is the homologous pair of chromosome 1. All chromosomes in human have homologue except sex chromosomes. Means all autosomes have homologous chromosome, which we will see later. Okay, so study about all about the chromosomes, like structure of chromosome, function, pathology, and behavior, is called cytogenetics. The cytogenetic study is done mainly by two methods, karyotyping and fluorescent in situ hybridization. So in this video, we will learn about the karyotyping and fluorescent in situ hybridization. That is the another topic. So we will discuss karyotyping. Okay. Now, next question comes in mind is how can we see these chromosomes and at which stage of cell cycle? So idea is to arrest cell cycle at metaphase and see through microscope. But why metaphase? We can see it in this slide. These are the stages of cell cycle. And as we can see, size of chromosomes vary in different stages of cycle. Like in interphase, chromosomes are thin and longer. In prophase, they are relatively smaller and thicker. Then comes metaphase, in which chromosomes are very thick, quite short, and well spreaded. Then comes anaphase. In this again, they are very small. So metaphase is the best to study chromosomes. So this is the metaphase spread, out of which we analyze the chromosome and make the karyogram. So in laboratory, we have to prepare this metaphase spread, for which we have to arrest cell cycle at metaphase by adding colcimide, which is a chemical which prevents spindle fiber formation during the cell cycle, and take that on slide, make chromosomes banded, and see them through microscope. Banding is the staining of chromosomes by chemical like GeneVistaByDrEkta so that we can see the chromosomes in microscope. After capturing this metaphase, we can analyze the chromosomes and make a karyogram. Finally, now we are able to detect any abnormality in chromosomes. That is our target. So, now coming to the main part of this video, that is chromosome identification. Before understanding the identification, we should know about the four categories of chromosomes, which are based on the position of centromere. So they are of four types. Metacentric chromosomes. When centromere is found exact at the middle of chromosome, so that two arms of chromosome P and Q are equal. Submetacentric chromosome. When centromere is present near the middle but not exactly middle, so both arms of chromosomes are unequal. Acrocentric, when chromosome is present at not present at center but near the end of chromosome, that is acrocentric. And finally telocentric, when centromere is located at the end of chromosome. Now, in humans, all 23 pair of chromosomes are classified into 7 groups A to G based on their size, centromere position, and banding pattern. Banding pattern, as we discussed, are bright and dark transverse bands in chromosomes. Before seeing through microscope, chromosome or metaphase spread should be stained with banding material like GeneVistaByDrEkta or any other. So on the basis of that, these 23 pair of chromosomes are divided into these 7 groups. So group A includes metacentric and submetacentric chromosomes, and it has 1, 2, and 3 chromosomes. Then group B comes, it includes chromosome 4 and 5, both are submetacentric. Chromosomes 6 to 12 comes under C group and all are submetacentric again. Then group D, which includes chromosome 13, 14, and 15, and these are acrocentric. Then group E, it includes chromosome 16, 17, and 18, and all are submetacentric. Then group F, which includes metacentric chromosomes 19 and 20, Chromosome 21 and 22 are included in group G. And lastly, sex chromosome X, which is a C group chromosome, and Y, which is a G group chromosome. Now, one by one, we will discuss about all the chromosomes in detail. So first comes A group, which includes chromosome 1, 2, and 3. Okay, to identify chromosome 1, it is very easy to identify. It is the longest metacentric chromosome. Longest metacentric chromosome. Although that is enough, but additionally, as we can see, there is a pale window at telomere part at P arm. Okay, then for chromosome 2, it is longest submetacentric chromosome. So basically for these two chromosomes, we have to see which is the longest metacentric, that will be chromosome 1, and longest submetacentric, that will be chromosome 2. Now chromosome 3. It is the second longest metacentric chromosome, but the main thing about this chromosome is that there is a crisscross bending pattern at centromere. This crisscross bending pattern at centromere is the main identity of chromosome 3. Okay. Now coming to B group, which includes chromosome 4 and 5. Both are submetacentric. Then to distinguish chromosome 4 and 5, we have to remember two things. There is dark shoulder band on chromosome 4, but no dark shoulder band is present on chromosome 5. Second thing, there is light cap on telomere part of P arm, and there is light cap on telomere of Q arm in chromosome 5. So chromosome 4 has shoulder band, but 5 doesn't have it. Chromosome 4 has light cap on P arm, but 5 has light cap on Q arm. So chromosome 1 to 5 are sorted. Pretty easy. Then comes group C. Okay, it includes chromosomes 6 to 12. All chromosomes of this group are submetacentric. So first chromosome is chromosome 6. If we see, there is a pale window on P arm between two bands. Okay. Unlike chromosome 1, 4, and 5, this pale window is not terminal. We can see in chromosome 1, 4, and 5, the pale window was terminal. But in chromosome 6, it is not terminal. It is present between two dark bands. And also, there is high density dark bands on Q arm. So, that is its identity. Then comes chromosome 7. It is also submetacentric. And main identity of this chromosomes is presence of snake eye. If we see, these are the snake-like eyes present on telomere side of P arm at chromosome 7. Although that is enough to identify, but additionally, there is presence of two dark bands on Q arm. So we can also say that chromosome 7 has distinctive three-banded appearance. Okay, chromosome 8, again submetacentric, and there is prominent dark band near the Q arm telomere and very small light window in the middle of P arm. This is. Okay, I will repeat. There is prominent dark band near the Q arm telomere and a very small light window in the middle of P arm. That is chromosome 8. Now chromosome 9. We can say the miniature of chromosome 7 as it is having three-banded appearance. distinctive three-banded appearance on it. Also, there is very pure light band at bottom of QR. So this is chromosome 9. Now chromosome 10, it is similar to chromosome 8 if we remember. In chromosome 8, there is dark band near telomere, while in chromosome 10, there is dark band near centromere at QR. Or another thing is, there are 3 dark bands evenly spaced on Q arm. These 3 dark bands evenly spaced on Q arm. This is the identity of chromosome 10. Then chromosome 11. Actually, chromosome 11 and 12 have 2 things to distinguish them. In chromosome 11, there is dark thick band near telomere, and on chromosome 12, there is dark thick band near the centromere. This is the dark thick band. This is near the telomere on chromosome 11, and this thick dark band is near the centromere on chromosome 12. In both the chromosome, there is light window between the large dark band and centromere, but this light window is large in chromosome 11 and it is smaller in chromosome 12, as this dark band is closer to centromere on chromosome 12. Okay. Now coming to D group, which includes chromosome 13, 14 and 15. All are acrocentric chromosomes. Actually, they are very easy to identify. In chromosome 13, there are 2 dark bands in the lower half of Q arm, while in chromosome 14, there are 2 dark bands but largely spaced. Means space between 2 dark bands is larger in chromosome 14, while it is lesser in chromosome 13. In chromosome 15, there is 1 dark band right at the middle of chromosome. That is it. The next group, that is E group, it includes chromosome 16, 17 and 18. First, to identify chromosome 16, it is metacentric and it has dark centromere and look like bow tie. It is bow tie shaped chromosome. That is sufficient, but if we see, there is dark band on Q arm here. This is the dark band, this is another dark band. So we can say Q arm is little darker than the P arm. So next, chromosome 17, which is submetacentric, and it has proximal dark band near the end of Q arm. That is its identity. Then comes chromosome 18. It is the bottle-shaped chromosome. Bottle-shaped chromosome, and it is submetacentric. Very easy to recognize. Also, there are 2 prominent bands near the centromere and another at telomere. There are 2 dark bands in chromosome 18 on Q arm, of which one is near the centromere and another near the telomere. The next group, that is F group, which includes chromosome 19 and 20. Both are metacentric. Like chromosome 16, this is also bow tie shaped chromosome with dark centromere, but obviously it is smaller than chromosome 16. And main thing to identify this chromosome is that it is totally pale chromosome. There are no dark bands on P arm or Q arm. So let's come to chromosome 20, which is also metacentric, and it is not pale. It is not pale chromosome. It has 2 dark bands, both near the tip of the arms, at P arm and another at Q arm. So that is the difference between these two chromosomes. It is not totally pale, but it is pale chromosome. Okay, then coming to G group, that is the last group, which includes chromosome 21 and 22. Both are acrocentric chromosome, but centromere in chromosome 21 is scattered. It is scattered here, but on chromosome 22, the centromere is dot-shaped, not scattered. Okay. Then the last group, sex chromosomes. Chromosome X, it is a C group chromosome and it is submetacentric. Main identification of this chromosome is presence of equidistant bands. Means there are two bands, one at P arm and another at Q arm, and both are at equal distance from the centromere. Both are at equal distance from centromere. So then comes last chromosome, that is Y chromosome. It is the smallest acrocentric chromosome. That is enough. So we have to see which is the smallest acrocentric chromosome. That will be Y chromosome. But additionally, if we see, there is dark band near the end of Q arm and dark cap on P arm. So this is the metaphase spread. If you want, I can add some metaphases in my description box. Okay, that was all about the identification of chromosomes. I hope it will help you guys. I'm pretty sure after doing some practice, you will be able to identify chromosomes very easily. Still, if you have any doubts, please feel free to ask me in comment box. And lastly, yes, this video is only for educational purposes. Do not use it in diagnosis without medical expert advice. I repeat, do not use this video in diagnosis without the medical advice of any expert. So, thank you very much. See you soon.